This is a recording of the Chapter 1 lecture we missed because of the snow day cancellation. So we're not reading all of Chapter 1, we're just picking up the parts that are relevant to studying evolution and taxonomy, which is classification of living organisms, and a little bit on scientific methodology because we always need to review that. So we're going to start with how living things are the same, that's the unity of life, and then we'll talk a little bit about how they're different, that's the diversity of life. So all living organisms, all of them, any trace of any living organism that we have ever found uses DNA as its genetic material. You probably know that DNA makes up the genes and chromosomes. The complete name of DNA is deoxyribonucleic acid. You're never going to have to memorize how to spell that, but you do need to have some basic concept of what it does. DNA determines which proteins are manufactured in a specific cell, and then those proteins go on to guide everything about that cell, its structure, its function, how it develops, how it grows, whether or not it gets to reproduce. So the specific proteins found in a liver cell in your body, for example, are going to be different than the proteins found in the retina of your eye. And though that protein synthesis is directed by the DNA. So DNA is like the executive in your cells. But DNA has characteristics that can be analyzed, and that is in the sequence of the nucleotides, the adenine, guanine, cytosine, and thymine that make up the DNA. It's a code, and like any code, there are going to be some sequences that you can analyze that are exactly the same from one organism to the next, and places where they are different. The more similar two organisms' DNA is, the more recent their common ancestor. So just as an example, knowing that DNA is inherited from parent to offspring, and chromosomes and genes, of course, are the structures that provide that inheritable units of DNA. So you got half of your DNA from your mother and half from your father. If you have a sibling, a brother or sister who's not a twin, then they also got half randomly from mom and half randomly from dad, and you probably have 50% of your DNA in common with a brother or sister. Now, of course, if you have an identical twin, then you're exactly the same in terms of your DNA. So analyzing the DNA allows us to understand how recent a common ancestor is, but understanding the role of DNA, that is, that it guides protein synthesis and that directs what cells look like, allows us to make a connection between the DNA sequences, that's your inheritance, and what you actually look like, what your traits are. So when we're looking at organisms to look at their traits, it's sort of a proxy for understanding what the DNA has in common. So how living things differ? Well, that's the traits, the observable characteristics. So even slight differences in DNA can lead to really visible changes. For example, a person who's albino can't make the protein melanin, which is the pigment molecule in our bodies. That's a very small difference. It could be just like one or two little changes in the DNA, and that's a huge difference in the observable characteristic. Sometimes those changes in the DNA can be beneficial. They can make an enzyme work faster or at a different temperature, or make the visual cells, the photoreceptors in your eyes, be able to see a frequency of light that you couldn't see before. So these differences can accumulate over time if they're advantageous, and that then creates more and more differences as different interacting groups no longer share their inheritance. And so the more distance your relationship, the more difference there's going to be, and you're going to see increasing scope of variation among living organisms. That's an increase in biodiversity. So you're going to have differences within a species, that's intraspecific, and differences between species, which will be greater than the differences within a species. So intraspecific versus interspecific differences are just part of the concept of biodiversity. So how do we kind of encompass this? How do we get our heads wrapped around it? We use different classification schemes to organize that idea of biodiversity, that variation. And so we do this at different levels, and one of the biggest levels is looking at the cells. Do they have a nucleus or not? We'll look at that again in just a sec. And then after you've sort of winnowed that down, between humans and chimpanzees, we're looking at 98% or more of our DNA is identical. But we're looking at levels of relatedness at narrower and narrower and narrower scales. So you are closely related to a brother or sister. You're a little more distantly related to your cousins. And that's kind of the same concept as what we use in classifying species. All right, so we're going to start with just an overview of the classification scheme that we use today. The highest level of classification, that is the broadest, biggest categories, we call domains. And there are 3 domains: the bacteria, the archaea, and the eukarya. Now, we used to have a different concept, and that was prokaryotes versus eukaryotes. And prokaryotes were thought to all be bacteria. And this idea of bacteria being teeny tiny cells with no nucleus, they have no membrane around their DNA, it still exists. I mean, it's still a fact that bacteria and archaea are prokaryotes. We just don't use that term taxonomically anymore. It's just a descriptive term. So bacteria are prokaryotic, archaea are prokaryotic, eukaryotes, eukarya, are specifically those things that are eukaryotic. So if it's a cell with a nucleus, that is the DNA contained within a membrane, then it belongs in the domain eukarya. And additionally, you see in these eukaryotic cells that there's more to it than just that. There are membrane-bounded organelles like mitochondria and endoplasmic reticulum, for example. All right, so when you look at bacteria, there's a lot of diversity in the bacterial domain. So you have cells that are kind of squiggly looking, some that are long, some that are short, some that are round, some that form chains, some that form spirals. And the identification of bacteria, the classification of bacteria is way beyond the scope of this course. We offer an entire course just in microbiology. In fact, it's an entire discipline of biology to study bacteria. So for now, that's enough. The archaea, like the bacteria, are little single-celled organisms with just a simple membrane around the outside and some chromosome on the inside. So they resemble bacteria, but if you look at their genes, if you look at the genetics of archaea, their genes are more similar to our genes. And we'll be looking at specific examples of that when we get more into the different groups. But just looking at the pictures of them, you wouldn't be able to say much about them. So the bacteria and the archaea, we're not going to discuss so much in terms of what they look like, because they're much more diverse in their metabolism and in their genetic makeup than they are in their appearance. Your book will tell you that these are typically extreme environment organisms. On the left in this picture, one that grows in sulfur hot springs. On the right, from seafloor hydrothermal vent, very extreme environments. But we do find archaea found in perfectly ordinary environments. In fact, you have archaea living in your gut. So I don't want you to be thinking of archaea as being just extreme organisms. All right, moving on to the eukaryotes. This is where we fit in, but not just us. So within the domain eukarya, the highest level of classification is a kingdom. And there are 4 kingdoms within the eukaryotes, one of which we could argue about. So the protists are so extremely variable that people argue about whether they belong in a single group or not. For our purposes, for simplicity, we're just going to lump them all together. The kingdom fungi includes everything that's a fungus, including mushrooms. The kingdom plantae includes most of the photosynthetic organisms on land. So that excludes the algae, because algae mostly live in water, and also because algae actually belong in the protist group. And then the last kingdom is the kingdom Animalia. That's us. So what we have going for us, I mean, obviously we're animated. That's why they call us animals. because we move, and plants are rooted in the ground. So that's kind of the underlying difference that give us the name. We are multi-celled consumers. That is, we don't make our own food through photosynthesis. Okay, so just a quick tour through the different groups, but we'll spend a separate chapter on each one of these during the semester. So here's our Protists group. It includes amoebas, it includes little single-celled, shelled marine organisms called diatoms and foraminifera and radiolarians. They're really cool. It can include some parasites, including parasites in human blood. And then in the picture on the left, what you're seeing is the giant seaweed called kelp. And kelp is considered a protist. So not all protists are single-celled and small. Kelp are really quite large. All right, in the fungi, you've got something as different as yeast and mushrooms. Fungi are usually saprophytes. That means they break down dead stuff. They don't have roots, but it sure looks sort of like mushrooms have a root, and so these used to be classified with the plants until we figured out not only because they're not photosynthetic are they very different from plants. In fact, fungi are more closely related to animals than they are to plants. So in the plant world, you've got things that are photosynthetic. They have green chloroplasts that can harvest energy from sunlight and turn it into sugar. And then finally the animal kingdom, which is where you will find us along with other mammals and insects and crustaceans and primitive things like sponges and jellyfish. Okay, so as we try to organize this information about species, we need to know how we use the term species, and we need to know how we classify species. So there are different ways to define the term species. For us as biologists, species can interbreed. That is, members of the same species can breed with each other, and the offspring will be members of the same species. That's basically a biological idea. But geologists also talk about species of fossils. And of course, fossils are not alive, so you can't exactly test whether they can interbreed or not. Furthermore, as we become more sophisticated in our study of DNA, we find that even though two groups could interbreed, well, maybe they don't in real life, and so we are changing our definition of species. It's really not an easily defined word. But for our purposes, two specimens are the same species if they could interbreed and produce a viable offspring of the same species. So we'll go with that definition for now, and we'll try not to worry about it too much. Our study of classification is called taxonomy. Each class, each level, like domain or kingdom, is called a taxon. So species is a taxon, and kingdom is just a bigger taxon. So taxonomy was founded by a Swedish naturalist named Carolus Linnaeus. Actually, that's the Latinization of his name. And we call the system he developed the Linnaean system. Linnaeus developed a system in which each different species had a two-part name. They were all given a genus name, that's sort of their family group, and then a specific epithet. Now, in this case, an epithet does not mean a bad word. It just means a descriptor, so the species name. So an example of this, you see the scientific name Panthera leo. Once you know what the genus name is, that is after you've referenced it or spelled it out once, you can abbreviate that. So you know that members of our species are Homo sapiens. And so we would write that capital H period and spell out sapiens after that. How do you decide if two things are in the same species or not? You're looking for shared inherited characteristics. But finding those and understanding whether they're shared or not and whether they're inherited or not becomes problematic. And as we get into our example of classification later on, you'll see just how problematic it can be. All right, so here's an example of 5 species being classified. These are species of flowers in the rose family, or most of them are in the rose family. Let's start with the far right. You see the dog rose, and a little pinker to the left, the prickly rose, and then the apple. I don't know if you knew that apples were in the rose family. And then marijuana, which not legal in Texas, so we won't be examining specimens in person. And then finally to the far left, the wild carrot. And obviously the three flowers that are all in the rose family, you can see the similarities in the number of petals and the arrangement of the flowers. So let's look down the list. They're all in the domain Eukarya. They're all in the kingdom Plantae. They're all in the same phylum, Magnoliophyta, which is named for the magnolia tree, and they're all in the same class. When you get to the level of order, you see that the wild carrot is in the order Apiales, and all of the other are in the order Rosales, and so that makes the carrot what we call an outgroup. The others are all sister groups at the level of order. Now, as you go down a level to family, you see that the marijuana plant is in a different family than the others. The others are all in the rose family. So that makes marijuana the out group for the three sister groups of apple, prickly rose, and dog rose. At the genus level, apple is in the genus Malus, whereas prickly rose and dog rose are both in the Rosa genus. So that makes the apple the out group for the two sister groups of the rose. And they're both in the same genus, they just happen to be different species. So you could say they are sister species. Now, as we look at higher levels of classification, what you're seeing here is two different ways of classifying things. Now, I'm going to tell you the one at the top, the Six Kingdom classification, is already considered out of date. All right, but let's follow the lines. I want you to follow the lines. So let me see if I can get my cursor on the screen invisible. Hoping this is going to show up. So here you see the ancestor, and here you see bacteria branching off first to the left. So starting at the bottom, the first line that branches off is bacteria. The next one that branches off is Archaea. Now look at the Protists group, and notice it's not one, it's 1, 2, 3, 4, 5, 6, at least 7 different groups of Protists, all of them apparently having separate origin. So to call all of that one kingdom is really not scientifically valid, because each one in and of itself ought to be a separate kingdom if they are that different from each other. So the term protist is really taxonomically not a very useful term. It's just like prokaryote. It's not a taxonomic term, it's just a descriptive term. And then we have plants coming off, and then fungi and animals being most closely related to each other. Now, in the bottom, all you see is the bacterial, Archaea, and Eukarya domains. And notice again that bacteria branch off with greater distance than the Archaea and the Eukarya, showing you that Archaea and Eukarya are more closely related to each other than they are to bacteria. All right. Concept break. If you want to pause, this is where to do it, but you can just come back and finish the recording. We're going to come back to this idea of science as a way of knowing, and instead of talking about it in more philosophical terms or big ideas, we're actually going to get into some nitty-gritty as we look into these methods. So first off, science of nature is mostly just organized critical thinking. A lawyer uses critical thinking. A plumber uses critical thinking. So critical thinking is simply a matter of making judgments based on the information that you have received. You're going to consider any evidence, any bias, any alternative interpretations. Anytime you're problem solving, you're doing this. So, but in science, we use what we like to call the scientific method. I'm going to tell you there's really more than one scientific method. There are lots of different ways of going at this, but generally speaking, if you're going to publish results, you need to include all these steps. So the first step is to come up with a hypothesis. Now, you can't just come up with this out of the blue. You have to have some prior information. Maybe you've observed something. Maybe it's based on an experiment you already did. But you're going to come up with something you can test. That is something you can devise an experiment or an observation on to show whether your explanation is valid or incorrect. Now, once you have that hypothesis, your idea that an explanation can be tested, you're going to make predictions. If my hypothesis is true, when I run an experiment, here's what is going to happen. And if your predictions do not come true, then that can disprove your hypothesis. So you can test predictions on what we call a model. It could be an experiment, or you could go out and actually make observations. You're going to collect your results, that's your data, and form a conclusion. And your conclusion either supports the hypothesis or it doesn't. If it doesn't, you reject that hypothesis, and then maybe you circle around and formulate a new hypothesis. The last step, the reporting the results to the scientific community, is how science accumulates knowledge. And it's crucial to the development of valid science to make sure all these steps are done correctly and properly, and that the results are reported. Okay, so where students tend to kind of mess things up is not in the data collection, it's in the data analysis. So we need to look at some of the main problems that occur in analyzing data. Now we're actually going to do a little bit of data analysis in class in a couple of different ways. I'm hoping you're going to watch this video before we get to that. So let's start with sampling error. Say you were testing the efficacy of a new drug. You want to see if a drug cures baldness. Well, if you start with a very small subset, maybe you start with just 10 people that you're testing this on, then understanding the idea of variability within a species, you're not getting a good cross-section of the variability of the human population. You have to look at factors such as age and sex and physical health and diet, and all of those things can lead to a sampling error if you're not sampling a large enough group. So to reduce sampling error, you want to begin with a larger sample. You want to repeat your experiments, keeping the conditions the same, to see if you get the same results every time. Let me give you an example of that. There was a paper that came out in the last week or so that was testing some chemical to see if it was effective in treating mice for some memory deficit, I think is what it was. And what they found was, if the lab technician who administered the drug to these mice, they're just in cages, if the experimenter was a female, they got different results than if the experimenter was a male. So somehow these mice were responding differently to female humans than they were to male humans. That's a really weird sampling error, but, well, it's actually a technique error, but it's an error. Okay, the probability, you're measuring the chance that a particular outcome will occur, and so we would say if we did a good job and we got good data, we might say we're 95 to 99% sure that our treatment had an effect. Okay, so here's a little demonstration of how sampling size how sample size affects sampling error. And here you can kind of see, this is out of your textbook. We have a jar with mixed green and black jelly beans. Our subject can't see, so she does not know. She doesn't know how many jelly beans are of each kind, so she's just doing a random sample. If she just samples one, the way you see at the bottom left, if she saw that, she would assume naturally that there are green jelly beans in the jar. So as you get down to the bottom right, though, you get a more representative sample. You get, generally speaking, a ratio of your sample of green to black jelly beans that's the same as the ratio in the actual population in the jar. So the smaller the sample size, the greater the error is likely to be. Now, sampling error, sample size are not the only thing that affects statistical significance. Statistical significance is simply a measure of how likely it is that the sample you took is a faithful representation of the actual whole population. So statistical significance doesn't measure whether the results are important or useful. It just measures whether it's the same in your sample as it is in the population at large. And sometimes there's sampling bias. That's why we blindfolded our subject in the previous experiment. And sometimes there are errors creeping in, like the fact that who knew that female experimenters would have a different effect on mice than male experimenters, that the scientific community should test the results. Unfortunately, between not enough resources and not enough time, very few reported results are ever tested in this way. All right, so that's just a little bit about scientific method. In your review questions, you have some terms that you need to define, and we'll be going over these a little bit in class. We're going to talk last about the nature of science. These terms get confusing sometimes, so we need to distinguish between a scientific theory and a law of nature. A scientific theory is an explanation. It explains how it is that a natural phenomena occurs, or what are the rules by which it operates. It has to be consistent with all the data gathered. If there are any data that's inconsistent with it, then that theory has to be either modified or thrown out. So a scientific theory is basically an explanation of some phenomenon, and those phenomena, if they're repeated events, get kind of promoted into laws of nature. So it's a law of nature that the planets orbit around the sun. The mechanism of that phenomenon, of that revolution, is simply the law of gravity. So we understand that gravity is a law. It's a phenomenon that occurs, but that doesn't explain the mechanism of how gravity works. So we have to have a theory, a scientific theory of gravitational effect, along with having a law that simply states gravity is real. So what kinds of things are science unable to address? If it's a question of philosophy, it's a matter of opinion, or if it is anything that is not within the natural world, then if it can't be tested, then it's beyond the nature of science. That doesn't mean it's not important. It doesn't mean it's not real. It just means it can't be scientifically tested. So science is limited to testing phenomena in the natural world, and any supernatural explanations of how life came to be, or why we're here, or why am I alive, those are all important questions. They just don't have a scientific answer. Science helps us communicate with our peers, with each other. It is a way for us to verify that things in the natural world really do exist. And it's our way to confirm our own experiences and share those experiences with our neighbors without being fooled into thinking something is true when it isn't. So that's the nature of the course, and that's the end of our lecture, and I will see you in class next time.